So today I want to talk about the project that we've been working on the last several years, where we've been trying to find a novel activator of X chromosome inactivation, and we think we have found one, and that is HFC1. So, as you know, female cells, to compensate for the extra dosage of X-linked gene expression, silence one of the X chromosomes by the process called X chromosome inactivation. And this is achieved mainly by tipping the balance between a set of autosomally encoded factors and X-linked factors. And it's been shown by many groups in the last more than 20 years that the pluripotency factor network, which is autosomally encoded, silences the expression of X's, the long non-coding RNA, in pluripotency. And then when cells will start differentiating, when these embryonic stem cells will start differentiating, these pluripotency factors are downregulated, and then a set of activators come in to kick off, let's say, the expression of X's that leads to monoallelic upregulation of one of the alleles. And in our group, for the more than 10 years, we've been working with an X-linked protein or gene called it's a U3 ubiquitin ligase, RNF12, which is actually situated here. And we've shown that RNF12 sends REX1 for degradation, and REX1 is one of the repressors of X's. It's a pluripotency factor. So in this way, we found that RNF12 is an indirect activator of X chromosome inactivation. Indeed, we showed several years ago that when we generate full knockout of RNF12, we see that the cells cannot upregulate X's in vitro. But then when we knock out its target gene, REX1, we see a rescue of the phenotype. So the cells can re-express X expression and lead to X chromosome inactivation. We show these in vitro, but we also show these rescue in mice. So these mice were alive. So this is indicated to us that there might be additional activators of X that might be X-linked. Additionally, we also showed that RNF12 heterozygote cells, they also, even though they're technically like male cells with one copy of RNF12, they can also inactivate the X chromosome, albeit at lower levels compared to wild type. So again, this told us that there might be additional activators on the X chromosome. And this is what we set out to answer, to find new activators. And this work was spearheaded by two great PhD students, Jeffrey Buren and Beatrice Stan. Jeffrey was mostly focused on the wet lab work and Beatrice on the bioinformatics work. So what we decided to do is to generate long or incremental deletions of the X chromosome in the hope to find one of the activators within that deletion. And we generated these in heterozygous RNF12 cells. And as I showed you just before, these cells can inactivate the X chromosome, but at a reduced level, so they're a little bit sensitized. And we found that within one of these deletions, deletion E here, you can see that the expression of X dropped. So we thought there might be an activator within this region. We then started generating overlapping and convergent deletions, and we showed that if we delete this deletion E to B, for example, shows the same phenotype as the deletion E, and then E3A, which is here on the and E3B, which is here on the right side, also shows the same phenotype. But E2A and E3A do not show the phenotype. So this is E2A and E3A. They don't show the phenotype. So then we came to the conclusion that in this small overlapping region, we might have the activator that we were looking for. By analyzing data sets that were published, we saw that the expression of HFC1 increases just after the onset of differentiation. And this does not happen for IRAC1. So we imagined, and this behavior of upregulation of HFC1 in these first days of differentiation is very similar to what we've seen with RNF12. So we postulated that HFC1 might be the factor that we were looking for, basically. So we tagged HFC1 with a FKBP and homozygously. And here you can see a Western blot that shows a very clear and neat downregulate or like a loss of HCFC1 expression after a couple of hours. And we basically confirm these results by treating the cells, differentiating the cells for a few days, 3 days, and then the last 24 or 48 hours, we saw that by removing HCFC1, we can also see these loss of cyst expression, as happened with the deletions before. And these happen at the RNA level, but also by RNA fish, where we see a loss of exist clouds in these cells, and it's quantified there on the right. So we send these samples for our RNA sequencing, and we also noticed that JPX and FTX, so two activators of exist, are also downregulated, as you can see here, although T6 and TSX are not. And we, as expected, from a phenotype of loss of exist, we saw that the X chromosome was not silenced, basically, as was expected. So what is the mechanism? So these indicated to us that HCFC1 might be our potential activator of exist expression, but what is the actual mechanism? So HCFC1 is a transcription coactivator. It was discovered in the herpes simplex virus, and it's a constitutively expressed protein, and it's critical for cell cycle progression. And this is one of the reasons why we did the conditional knockout with the FKBP, because homozygote knockouts are basically lethal. And it's been shown to tether chromatin modifying enzymes, such as for the deposition, for example, of A3K4 trimethyl. It has several domains in the protein, and one important domain is a pro domain that you can see here, and that's where the protein gets proteolytically cleaved by an enzyme called OGT, as you can see here. And funnily enough, OGT is also X-linked, which is quite interesting. And this leads then to cleavage of HCFC1, and these different fragments basically interact with each other for its function. Metadata analysis of its interactors has shown that HCFC1 is a component of the COMPAS complex, and the COMPAS complex is very important for the deposition of A3K4 trimethyl at promoters. But we also notice, and has also been published, that HCFC1 interacts with WABA1. So here on the right you can see the chipsets from publicly available data of HCFC1 on the left here. And then you can see A3K4 trimethyl deposition. You can see one of the subunits of the COMPAS complex, and also Pol2 serine 5. And you can see that they colocalize very neatly, so as was expected. But we can also see that there is some sort of a colocalization with WABA1. And these for us was actually quite interesting, because as has been shown by the group of Claire 10 years ago, if you knock down WABA1, you also lose cyst expression. So these, we hypothesized a mechanism where WABA1 might be recruiting HCFC1 to chromatin, because HCFC1 doesn't have a DNA binding domain. So it might be recruiting HCFC1 to chromatin, which might in its turn recruit COMPAS to deposit A3K4 trimethyl at exist locus, and then these would lead to activation of exist. So to confirm this interaction between WABA1 and HCFC1, we tagged WABA1 with a flag tag. We pulled it down, and we did mass spec on the interactome. And even though the interaction with HCFC1 was just below the significance level, we saw that WABA1 interacts with the ENO80 complex, but also, and importantly, with the integrator complex. And this is also something that has not been published. And it interacts with the core subunits of the integrator complex, which is also very important for transcription and release of post polymerase, basically, at promoters. So the question that we had is does HCFC1 mediate A3K4 trimethyl dependent cyst activation through WABA1 and integrator? So to do this, to be able to reply to this question, we tagged WABA1, in this case, we tagged WABA1 with an FKBP, basically the same system we use for the HCFC1. And then we confirmed that by removing WABA1, we also see this downregulation of exist, confirming the data from Claire's group 10 years ago. And we also see a downregulation of JPX and FTX, and a little bit also of T6. And so what we then decided to do is with these cells where we can remove at will WABA1, we chip-sect different either chromatin modifications or these two proteins, HCFC1 and integrator. And we saw that removal of WABA1 doesn't lead to major changes in H3K4 trimethyl deposition genome-wide. Around 7 to 8% of HCFC1 peaks are reduced. So it seems that to that subset of HCFC1 peaks, YY1 is recruiting HCFC1 to that. And this is actually known because HCFC1 has been also shown to interact with other transcription factors such as RONIN or ZNF143, GFY, etc. But then when we looked at the integrator binding to chromatin, we saw that around 1/5 of the binding sites of integrator, or one of the subunits, were reduced. So it seems that YY1 does recruit integrator to 1/4 of its binding sites. And then, so this is in the YY1 DTAC line, and we also checked the H3K4 trimethyl deposition in the HCFC1 DTAC line that I was just showing you before. And again, surprisingly, we see that there is not much change in H3K4 trimethyl deposition, even though HCFC1 is a component of the compass complex. So around 10% of the sites have reduced H3K4 trimethyl. So this is at the level of the genome, and the next question that we had is what's happening in the XIC, in the X inactivation center. So let me focus a little bit in these locus, and all of this data is shown is from day 3 of differentiation, except when it's written. So as you can see here on the top part, we see the H3K4 trimethyl deposition in the HCFC1 lines or in the YY1 lines. And as expected, we see a loss of H3K4 trimethyl in both the YY1 loss and the HCFC1 loss, as you can see here. But there's not much change at the level of H3K4 trimethyl in the JPX locus and FTX locus, even though we see a downregulation of their expression. So we see loss of a downregulation of their expression, but there's not that much change of H3K4 trimethyl at their promoters. And then we looked at HCFC1 binding in the YY1 lines, and we see that there is not much of HCFC1 binding actually at the cis promoter at day 3 of differentiation, but there is at day 0 of differentiation. So these are male lines that are from the ENCODE dataset, and you can see a little bit of HCFC1 binding. But at day 3, we don't see that much of a little bit of HCFC1 binding, but it does not seem to be dependent on YY1. However, when we look at JPX and FTX, we see that HCFC1 binds more strongly, and at least in JPX, we see that the binding is YY1 dependent. But that's not the case basically with FTX. And finally, when we look at integrator binding, we see that there is not much integrator, or there is a little bit of integrator binding to the cis regulatory region, but that doesn't seem to be dependent on YY1. But if we look at, again, FTX and GPX and FTX, we see that integrator does bind and seems to be a little bit reduced, which might explain why we see a loss of expression of these two non-coding RNAs, even though H3K4 trimethyl is unmodified. And we also see these happening at the level of T6. So a complex model arises where HCFC1 might be recruited compass to install H3K4 trimethyl at the cis locus, but at the level of which would lead then to activation of exist expression upon differentiation. But at the level of JPX and FTX, this might happen through a different mechanism, which might be YY1 recruiting integrator, which will affect the transcription of these two genes, even though the chromatin modification are unchanged. So to conclude, we've done a forward genetics analysis to uncover a new activator on the X chromosome, and we think we found one, and that's HCFC1, which is a member of the compass complex. We see that YY1 recruits HCFC1 to a subset of genes, but also to 1/4 of the integrator sites genome-wide. And even though I haven't spoken about this, these seems to be independent of H3K4 trimethyl. And we also see that HCFC1 recruitment to exist seems to be YY1 independent, but it seems to be YY1 dependent in JPX. And we believe that, we see that the transcription of, or the expression of JPX and FTX might be regulated at the level of YY1 and integrator and less based on the H3K4 trimethyl mark present at their promoter. Several questions that we still have is we've been looking mostly at day 3 of differentiation, and the questions that we have is should we maybe look earlier at day 0 to see whether there might be differences there already, and whether the peak that was clearly seen at day 0 might be YY1 dependent. We're also looking into downstream effects of integrator removal at the level of JPX and FTX, and also we're investigating whether HCFC1 might have other partners basically at the level of the XIC. And with this, I would like to thank all the members, especially the ones in bold, Jeffrey and Beatrice, and then Evelyn and Samuel and Sarah for some work they've done on the Salons, and Christina for the mass spec, and of course Joost. Thank you very much.